So botulism, this was our objective for this one. It's generally not an infectious disease. What does that mean? It's not, let me put it this way. What typically, does anyone know, makes you really sick from the botulism organism? What's Botox? Everyone knows what Botox is nowadays, right? What is it? It's botulism toxin. They purposely inject into you, right, to deaden the nerves that control the muscles, okay, so that they don't contract, so you don't get wrinkles, right? Okay, so the problem here most of the time isn't necessarily an infection, in other words, the organism growing inside of you, but the toxins that it produces or that you may ingest all by itself. Right, so this one's typically referred to, a lot of times we worry about this one contaminating our food, right? It's one of the common foodborne illnesses. So people will experience blurred or double vision, weakness, nausea, vomiting, diarrhea, general paralysis, and respiratory insufficiency, right? Because as I said, it affects your motor neurons. So it's caused by a Clostridium botulinum. The bad news about this one, it's a spore former, right? So the spores can be in the environment. It grows anaerobically, so it has to be under anaerobic conditions. It is gram-positive, and it is rod-shaped. The problem is these endospores germinate in your food, right, and release a neurotoxin. The toxin is ingested. It survives stomach acid and enzymes and is absorbed by your small intestines and carried to the bloodstream to your motor neurons. It acts by blocking the transmission of nerve signals to the muscles, causing paralysis. So not all that different from when we talked about with tetanus. It can also colonize the intestines. So it can grow, you can have an infection of your intestines of this, although that's much more rare. Wounds also, again, could get contaminated, especially if you have an anaerobic type wound. How we talked about puncture wounds, right? Burns, contusions. And it can cause generalized weakness and paralysis. So again, most oftenly it's contaminated food. The most common offender is home canned foods that are not acidic and was not heated enough to kill the spores, which that's really difficult to do at home, to get the conditions such that you would destroy these spores. The spores are widespread in our environment, soil, aquatic sediments, and dust. Because of that, as I said, the intestines of adults, infants is really when you run into problems. And mostly when your normal flora have been disrupted, right? So again, this is more of an opportunistic pathogen. If you ingest the spores, right, they could germinate in the intestines. Wounds that contain dirt and dead tissue, again, that anaerobic environment, the dirt was probably the source of the spores. And again, injected drugs, you're creating a puncture wound. If that needle is not clean, you could introduce those spores into into your tissue. Education and proper home canning is very important. It is a heat-liable toxin, which means that if you do heat it, you can kill it. So home canned foods, if it's something you're going to reheat, then it's safer. Where jellies and jams and things like that that you don't normally reheat are more likely to cause problems if they're contaminated. Treatment, they're going to enemas and stomach wash, try and get the toxin out of your body. For wounds, we're going to do proper care, right? Remove the dead tissue, we're going to remove the dirt. They'll give you antitoxin. Remember, these are antibodies against the toxin. And depending if respiration has been affected, right, they may have to put you on an artificial respiration until they can get it cleared from the system. So there have been several outbreaks, as I said, home canned food as well as fermented foods that are very common in Alaska native populations, where they do these special ceremonies. And so you'll notice that one of them way back in, 80, I'm not sure what if each one of these tickers is a year, but back in the late 80s, right, there was an outbreak in Texas with some baked potatoes, right? A significant outbreak with chili sauce in Texas. And there have been numerous other outbreaks. Pruno, I don't know how you say that. I thought they had it defined here. Oh, I had looked it up at one time. It's some type of fermented something or other. Can't remember what. But there were several outbreaks due to that particular food source. It's not something I've ever heard of. Neither of any of y'all. No one's yelling out what it is. So the other thing that they take into account is, like I said, for infants, it can be quite serious, having it grow in the intestines. So as you can see, in 2012, we had quite a significant number of cases of this, of infants having this type of infection. So the next that we're going to look at is Hansens Disease, is the PC name, you could almost say, for leprosy. And there are some really distinctive features of this type of infection. And most people think of it as, it's been around since biblical times, right? People were sequestered and put in leper colonies for this. It still happens nowadays. People still get leprosy, but isn't quite the stigma it had in the past. Plus, we have drug treatment for it now, right? We do have antibiotics that can treat this infection. So skin lesions that lack sensation, and there's a significant reason why you have a loss of sensation, a loss of feeling. As well as deformity will sometimes happen to the face. Can even have loss of toes and fingers. And this is mostly due to the infection and the loss of sensitization that will happen. And then, you have some injury to the toe or the finger or something, and that is usually what results in the loss of that limb. So the causative agent is Mycobacteria leprae. It's an acid-fast organism, right? So one of the stains that helps in identifying is the acid-fast stain. It's non-culturable, which means that we cannot grow it in artificial culture, right? That has to be grown actually in an animal host. The animal hosts that they use for research, two of the animal hosts they use are nine-banded armadillos. Get the same infection that people do. And they also use nude mice, foot pads. They inject it into the foot pads of nude mice. The nude mice don't have any hair, and part of the reason for that is that their thymus is not functioning properly, which is also why they can grow this bacteria in very large numbers in the foot pads of these mice. These mice are kind of like the bubble boy, right? They're kept in a sterile environment. It's one of the ways that they'll propagate and grow the organism to be able to study it. So the problem with this one is that it invades the small nerves of the skin, right? So our peripheral nerves, right, of the skin. It multiplies also inside macrophages. So the cells are supposed to gobble it up and destroy it. So just like when we talked about Mycobacteria tuberculosis, same thing, right? They grow inside the macrophages. The course of the disease depends on the immune system response of the host. And this is one where it's really interesting because it really is dependent on the person. So remember, we have our cellular response, right, by T cells and cytotoxic T cells, and we have our humoral response, which is an antibody response. Now this is an intracellular bacteria, right? It can get inside, it can grow inside our macrophages. It attacks the peripheral nerves. So which type of response would you want? A cellular or an antibody response? What you think? What type of response would be best if we're talking about an intracellular parasite? The bacteria actually gets inside your cells. Are antibodies going to help you? A humoral response? No, those can't get inside those cells, right? It'll only help you when the bacteria is outside of the cells, which is a short period of time, right? Because it actually grows inside of our cells. So, humoral response, if your body goes just in that direction, that's the worst direction you could go in because you're not going to be able to kill the ones that get inside the cells. You want a cellular response, which is on the other end of the spectrum. I'm trying to remember if they put the terminology in here or not. I think I have it in later. Yeah, on the next slide. So your immune system is determined by your DNA, right? You're pre-programmed how your immune system is going to work and how it's going to communicate. So a lot of times you'll see that this disease kind of runs in families, and part of the reason for that is you're exposed to the same type of environment, right? So you probably pick up the organism, and your immune system is going to respond in the same way because genetically you're very similar to one another. Activated macrophages, right? So when we get helper T cell help, can limit the growth of the bacteria. But the problem too is that our nerve cells are getting infected, right? So when we have it, we do have a cellular response, right? Those cells are going to attack our nerve cells that are infected, right? So attack of immune cells against infected nerve cells causes the damage to the nerves. So then what do you have? Loss of feeling, right? This loss of sensation that happens. And this is typically what will lead to the deformities that we'll see in what's called lepromatous leprosy. And this is especially when you're mostly an antibody type response. Where you get that continued destruction, damage. Lymphocytes fail to react to the bacteria, allowing unrestrained growth of the mycobacteria. Direct contact, usually with the mucous membranes of an infected individual, is how you would come in contact with it. There's no vaccine. Treatment, there are several drugs that they use, and again, it depends on what type of leprosy you have, how the immune system is responding. Will determine the length. So notice months to years, individuals will have to take drugs to be able to eliminate the bacteria. And again, this has to do with the intercellular component. So multiple drugs for a long period of time. So early detectable findings in humans is the infection of the small nerves of the skin. Usually they're going to take a biopsy, right, and they're going to stain it and look at it and look for the acid-fast bacteria. It is the only known human pathogen that preferentially attacks our peripheral nerves. Right? Some people have peripheral nerve damage with diabetes, right? But that's not an infectious disease, right? It's the only known pathogen. Grows slowly, as I said, within our macrophages. The course of infection, as I said, really depends on your immune response. The macrophages can limit once they've been activated, but they've got to be activated to do that. The bad news is that, even if your cellular response is really good and you kill off all the bacteria were in your nerves, right? So you've damaged the nerves, and this type of repair cannot be fixed. And this typically happens in what we call tubercular leprosy, and this they say is rarely transmitted to others. But again, when we talk about transmission, and the type of leprosy you have, they really shouldn't be linked, because the type of leprosy you have is really dependent on your personal immune system, right? What they mean by that is that these people's immune system usually aggressively attacks and will hopefully kill off the bacteria, so they're less likely to spread it to others. Does that make sense to y'all? Okay, so the problem with this one is this is the one where you end up with a lot of the deformities, and part of the reason for that and why it's the fingers and toes and the face, nose and ears and such, this bacteria actually likes much cooler than our core body temperature, likes much cooler temperatures. And that's why it grows so well in the 9-banded armadillo. They're mammals, just like us, but their core body temperature is about, let's see, we're about 37, and I want to say they're around 34. I mean, they're significantly like 3 degrees cooler than we are. Where for us, that's our core body temperature, but at your extremities, you're cooler. And so that's probably, again, why they prefer the peripheral nerves, right, and why they're on the periphery is because it's much cooler there, right? The problem is that, you know, the nerve damage that happens, you don't feel sensation. And so I used to work in leprosy research at LSU Vet School when I first moved to Louisiana. And they have a museum still out at Carville, which is just south of Baton Rouge in St. Gabriel Parish. And there used to be a leper colony there. That was a group of lepers that were taken actually from New Orleans up the river and settled in a plantation there by a group of nuns. And that has been discontinued, but I mean, that was like the world center for research. And like I said, there's still about 100 new cases in the United States each year. It's a really serious problem apparently in India. Good sanitation for the most part is why we don't see it so much anymore, but and the fact that most people aren't necessarily even susceptible. Like you're able to very quickly and aggressively, if exposed, eliminate it without getting to the disease state. But I was told a story by one of the patients there that he had put his hand on a hot plate in one of the other patient's rooms, and he didn't know it was burning, that the hot plate was still on, because he couldn't feel it until he smelt his burning flesh, right? So just like diabetic patients and stuff, where they get the loss of sensation in their feet, the same thing for them. I mean, even though they've been cured, right, we have drug treatment for it now, we can't fix the nerve damage that has happened. So they have to make sure that they keep an eye on themselves, make sure they don't have any minor cuts or wounds, right, that could really get really bad. I mean, you know, when you cut yourself, you feel it, right? You're like, ouch, right? They won't, right? They won't feel it. I mean, literally will burn their hand and not even know it until they smelled it, right? And that's a very serious infection. I mean, you could definitely lose your hand from that. And so that's what would really happen to them, is they'd get secondary type infections because of, you know, wounds that they would incur because they couldn't feel that they were whacking themselves on things. The other form is lepromatous leprosy, and this is where they tend to be more on they do more of an antibody type response, and this is bad because the bacteria can grow to very large numbers in the mucous membranes, and you get very little inflammation response, right? We're getting more of just an antibody response, and so that one tends to be more of the lethal form. And of course, more likely to spread it, right? Because it's multiplying in their mucous membranes. But so individuals, when you look at how their immune system responds, there are some that go all the way to one end of the spectrum, lepromatous, mostly antibody response, and others that will go to the tuberculoid. But there are some people that will be right in the middle. They'll both do an antibody response and a cellular response. Right, and so that would be good, right? And that's probably like most people that don't even realize they got sick. And then there's variations in the spectrum where you could be a little bit more on the antibody side but still have some cellular response, and some cellular response, large cellular with a little bit of antibody response, right? And that's what you would hope for in general, right? When you are responding, you're going to go at it full force, right? Cellular response, antibody response, throw everything you've got at it, right, to hopefully eliminate it. But the really unique thing about leprosy is this peripheral nerve attack, right? It's the only one that does that. And so as I said, we do have cases in the United States each year. They're usually around 100. We don't see quite the numbers we saw in the past. And I think it's just, you know, educating people that are in environments where it's present and within families. And there isn't the stigma that used to be, because we know what causes it now, right? And we don't have quite the deformities that people would have in the past because we catch it early enough. Hopefully with proper diagnosis. But a lot of the people that are still there, because that's the only place they've lived forever, although there's not a colony there anymore, there are still some patients there that have cottages near the museum. And we're talking about people in their 70s and 80s and been through before there was a cure. Quite impressive people. There's a gentleman there that's from the Philippines. I mean, I'm not kidding you. People came from all over the world to get treatment for this disease. So the next is the viral diseases of the nervous system. So the main cause agents for viral and meningitis, right? So there are several that are linked to viral meningitis. And these are small, non-enveloped RNA enteroviruses. So these are ones that tend to be ingested and affect the intestines. Usually Coxsackie or ecoviruses of this particular group. The mumps virus can cause meningitis, of course, if you haven't been immunized, right? So that's one of the fears with that one as well. So you're typically, when it's viral, you're going to have quite an abrupt onset, fever, severe headache, stiff neck. Remember I said that's one of the key things, is that really stiff neck where the individuals really can't move their neck. Often vomiting, sometimes sore throat, especially if it's mumps, right? You'll have the enlarged parotid salivatory gland near the ear and the cheek of the face, rash or chest pain. So viremia, which means that the virus has spread to the bloodstream. And that's how it actually gets to the meninges, right? Meningitis, remember, is an inflammation of those membranes surrounding your brain and spinal cord. When they look at this cerebral spinal fluid, how they can differentiate this from bacterial infections is that fewer leukocytes, fewer white blood cells are going to enter in the spinal fluid than you will see with bacterial infections. All right, so if it's a bacterial infection, you're going to usually see a lot of white blood cells, especially mononuclear ones, like phagocytic cells, right, monocytes, macrophages, right, mostly monocytes. With the virus, there's usually no decrease in the sugar level. That's another thing that they're going to look at. Because remember, viruses have to get inside of our cells to multiply, right? So they're getting inside the cells of that membrane. That's the reason for the damage. Where when you have a bacterial infection, right, and they get into this fluid, they're like, woohoo, food, right? They're going to eat that sugar. So you'll see a significant drop in the sugar, the glucose, in the spinal fluid if it is a bacterial infection. So those are two defining things. And then, of course, if it's bacterial, and if they look at this under the microscope to look for the white blood cells, what else might they see if it's a bacterial infection? The bacteria themselves, right? They'll see bacteria. Bacteria in your spinal fluid is not normal, right? So when you see bacteria, you see lots of monocytes, you know it's a bacterial infection, right? You have a decrease in the glucose. If you don't have those indicators, it's probably viral. And that's kind of bad news, right? Because we're limited in how we can treat viruses. So most commonly, how do you come in contact with these viruses? It's the fecal or oral route, right? You've ingested, unfortunately, some amount of fecal contamination. The mumps is respiratorily, though, and saliva. What's your best prevention with any disease, right? Hand washing, avoiding crowded swimming pools during epidemics, right? They'll shut them down when that happens. And of course, for the mumps, make sure you get vaccinated. For viral meningitis, no specific treatment, right? Because again, it's a virus. So we're going to support you, right? Supportive care until hopefully your immune system can take care of the virus. What's the difference between sporadic and epidemic viral encephalitis? So sporadic is usually due to herpes simplex viruses, but other viruses, including those that cause mumps, measles, and infectious mononucleosis. Epidemic viral encephalitis is usually caused by the arboviruses. So these are the ones that are transmitted to us via arthropods like mosquitoes, right? Biting insects, mites, ticks. Mosquitoes are a big one though for us. I don't know why it's not on the list. So again, infected mosquito, it has it here, introduces the virus, right, into us. The virus multiplies locally and then establishes a low level of virulemia. When we say that, we're referring to it as entered into the bloodstream. When it reaches the bloodstream, right, then it could possibly attack the blood-brain barrier and enter into the spinal fluid and cause inflammation. So destruction of brain tissue can cause death, permanent disability such as emotional instability, mental retardation, paralysis of face, arms, leg. Due to brief virulemia, there is no exit for the virus, thus humans are the final host. All right, so we're not transmitting it. So symptoms within the first few days. Often within a week. Usually caused by one of 4, and the list is longer now, of several different arboviruses that we'll look at. What's one on here that we talk about here in Louisiana that's not listed right here? They talked about it on the news this morning. Good old summer coming in the biting mosquitoes. West Nile, right? So this is an arbovirus. Replication of the virus is set at the site, right? And it's when it gets into the blood and then possibly cross the blood-brain barrier that you have a problem. The virus is transmitted from humans from birds or rodents by the mosquitoes. Chickens are also used as sentinels to help warn of epidemics, right? So they'll get it first typically, right? And they'll test for them. Here in Louisiana, they actually trap mosquitoes and test them for these viruses. And so if you live in an area where they spray for mosquitoes, if all of a sudden you seem like they're coming by more often, that means that they had a positive result in their traps. And so they're killing more mosquitoes. So relatively lower numbers of some of these, and notice that they're reporting neuroinvasive. So some people get these infections, they get flu-like symptoms, and they don't develop to the level where it invades into the nervous system, right? This is the scary level, neuroinvasive. So this is what they're keeping in track of in this particular chart. So the California serial group of viruses, right, you can see we've had some significant spikes, and this is for the entire United States, right, at one time. Eastern, the bottom one here is the Eastern equine encephalitis virus, and then the St. Louis. And as you can see, one of the reasons why I grabbed this chart from 2009 is that notice where the numbers occur. Of course, in the summer months, because it's transmitted by a mosquito, right? June, July, right? Those are our big months for these guys. And look for West Nile down here, right? Big numbers in August. So as I said, you know, over the years, not been so bad for some of these other ones, but we've seen significant increases, right, in the West Nile. And on this one, they actually have the non-neuroinvasive cases as opposed to the neuroinvasive cases. This is a more up-to-date one. I had left the old one. So they had a significant spike again in 2011 of the California. And then West Nile, again, unfortunately, because we have so many freaking mosquitoes in Louisiana, right, we tend to be high on the incidence rate. And again, this is for the neuroinvasive of West Nile. So that brings us to rabies. And how is it that rabies is so damaging? What does it have the ability to do? So how do you get infected with it, right? Usually it's due to a bite of a rabid animal. Usually bats are actually carriers, and most people don't realize that. And inhalation is another possible mode when you're dealing especially with like bat droppings and stuff like that, though I think most of us probably avoid that. Anyone know the major rabid animal problem in Louisiana, wild animal? Do not mess with them. They're cute. Look like little robbers. Raccoons. Raccoons. Stay away from raccoons in Louisiana. They're one of the major carriers. I'll tell you, if you bring a raccoon to LSU vet school, they don't rehabilitate or anything like that. They kill it immediately and test it for rabies because it's that endemic in that population. Okay? Leave them alone. Right? If they're out during the day or they're coming near you, just don't. Right? Just leave them be. If you have a serious problem, call animal control. Right? If you have them in your area. And then, of course, we vaccinate our companion animals to create a buffer zone between us and the wild animal population. There's no way we'd be able to vaccinate the entire wild animal population out there. But unfortunately, our companion animals may come in contact with wild animals and then therefore could transmit it to us, right? So we help protect ourselves. There is a vaccine for humans as well, especially ones that work with wild animals or work as a veterinarian or with a veterinarian. So there is an effective vaccine for it. The good news is that we know how you get it, right? So most people are able to avoid it. So the few people that do get it each year, it's so few that we don't know if what we're doing to treat is actually effective, because again, we're dealing with a virus, or if the person's immune system saved their ass, right? So, and I don't think any of us are lining up for rabies trials to find out what's the best way to treat this, right? I don't think we want to, I don't think that one we're okay with not knowing, right? How it is that we're protecting or how these people survive. So during the incubation period, the virus multiplies at the site of the bite, but this is the problem. Then it travels via the nerves to the central nervous system, right? And this is why it's so lethal and deadly for humans. Here it multiplies and spreads outward via multiple nerves to infect the heart and other organs, right? But it's the nervous system destruction that really will get you. So there's our cute little guy, right? Leave him be. It's a pretty striking, unique virus. It's got this unique bullet shape to it. And when they're looking for, and I can never find a good picture of this, but when the virus grows inside the nerve cells, it produces these these formations as the viruses are forming, like as they're replicating inside the cells, and it's referred to as Nigra bodies. And so this is a diagnostic tool when they're looking under the microscope at these cells. If they see this characteristic arrangement inside the cells, it's a good indicator that they're looking at rabies. Right? So there's a way microscopically that they can detect. So the last is looking at the pathogenicity for African sleeping sickness. And the good news for this, denoted by the name, do we have it here in the United States? No. Mainly it's a problem in Africa. And it was actually one of the main reasons why when I was in college I didn't go to Africa on a trip. And I really regret that now. But I learned about this and it terrified me. Don't let these things terrify you. Like on the news this morning, you know, they were talking about mosquitoes, and I didn't pull up the numbers for Louisiana, but obviously we have the more higher incidences of neuroinvasive cases of West Nile. But, you know, don't become a hermit. Just, you know, protect yourself. Put on bug spray. Be aware of the mosquitoes, right? Try to avoid when you can, and protect yourself. The chances of getting it are out there, but they're not, it's not huge. It's not like if you go outside and you get bit by one mosquito, you're gonna get this, right? Do you understand what I'm saying? And so don't become paranoid, right? I know sometimes like people are like, how do you teach this and how are you not paranoid? I got over that a long time ago is what I tell you guys, right? So unfortunately I panicked at this one time. So it's a bite of a particular fly, it's the tsetse fly that transmits, and this is actually a protozoan, right, that causes this infection. So it's a flagellated protozoan, Trypanosoma brucei, and there are actually two different subspecies of this one. So the protozoa multiply at the site of the bite, and then they enter into the blood and lymphatic circulation. As new cycles of the parasites are released, they change their surface proteins, right? And so basically, they have a life cycle, right? And so they go through this phase, right, where they're multiplying, and then a whole bunch of them come out at one time. The problem is they're different than the previous generation. Right, that generation time the time it takes for them to multiply. So then your immune system has to start all over again and find antibodies for that particular new change that they've done. Now the wild animals are the reservoir for the subspecies Rindinsia. So what happens is the wild animals there in Africa have this protozoa infection. The mosquitoes bite them, and then they bite us, right? And this is the worst infection. This one progresses the most. There is treatment. There are drugs that can be taken. But just like for us with the mosquitoes, the best thing is to prevent getting bit, right? Wearing special clothing, insecticides, clearing brush where flies breed, right, and insecticides for killing off them. So the first symptoms appear within a week after the bite from the tsetse fly. A nodule develops at the site of the wound, right? Regional lymph nodes enlarge. Symptoms may disappear spontaneously, right? The immune system's got to figure it out. But then you're going to get the next wave, right? Weeks or even years later, recurrent fevers develop. Central nervous system involvement is marked by gradual loss of interest in everything, marked by decreased activity and indifference to food. Eyelids droop, individual falls asleep during daily tasks, speech becomes like me, slurred, right? Followed by coma and death. And that's why it's called sleeping sickness, right? People basically progressively seem to just go to sleep. And so this is what the protozoa look like under the microscope. These little wiggly guys, they have flagella. As I said, there are two different species. The Rhodinsia occurs mainly in the cattle raising areas of East Africa. The Gambezi occur mainly in the forested areas of Central and West Africa. And this one, humans can be a reservoir for this, right? So it's transmitted from human to human. Because of that, though, the disease progression doesn't tend to be as severe as the case with the Gambizi, where it's coming from the wild animal population. Because if it progressed really quickly and the people died, then it would die as well too, right? Because you need the host to harbor the protozoa and for the tsetse flies in this case to pick it up from one person and transmit it to another. So, as we said, enters through the bite, multiplies in the skin. The body responds with fever and IgM antibodies, but it's got to do this each time, right? The parasitemia happens, the parasites found in the blood. So as I said, the Rhodinsia is not so good, right? You can get central nervous system involvement within 6 weeks and death within 6 months, where it's much slower with the Gambizia. And I think, yeah, we're going to end there today. So all we have left is blood and lymphatic, right, for next week. I'm thinking on Friday we might do a little bit of a fun activity I'm putting together for bonus points again. So definitely want to make sure that you're here.